We're going to get started with our next company presentation, Dr. Paul Blake, Chief Development Officer, to get us started on Oxford BioMedica. Paul?
Thank you, Josh, and thanks to the organizers for inviting us and giving us a chance to present here today. I tried to follow the rule of 2 slides, but we thought we should put the disclaimer up about the forward-looking statements and the uncertainties, and I actually have 3 slides. The first is an overview of the company. The second one will be about our pipeline, and the third one will be about our news flow this year and next year as we plan it. We've been in business, this is our 19th year now, based in Oxford in the UK. And we have 2 platforms from which we work, gene therapy and gene delivery platform based on lentivirus. And I know a lot of today's talking has been about AAV, but we're the lentiviral guys. And separately, which we won't spend much time on, is our 5T4 antigen cancer immunotherapy projects. We do have a pipeline of our own products, some of which we're trying to take forward ourselves, at least until we get a brilliant offer, and some of which we are all already partnered out on, and I'll give you some detail about those. Now, the business model, we hope to have 3 revenue streams. One is from our own products that we take to market ourselves, or from the licenses and royalties from potential partners. We think we have some strong IP backed up by a lot of know-how given the years in business we've had, and we get license revenues from those. And then we also do some manufacturing of products with milestones and royalties associated with them, and some of that you may have seen. So we think our intellectual property has been validated by the partnerships that we've already made and announced. The most recent one was with Novartis for CTL19, and that was talked about earlier today. We also have partnerships for the lentiviral platform with GlaxoSmithKline and with Sanofi. And indeed, Sanofi have taken licenses from 2 of the products we took into the clinic, one for Stargardt's disease and one for Usher's syndrome. And then we also have process development with both IP and a lot of know-how, because we were frustrated in using third-party manufacturers, and we decided we'd be better off doing this ourselves, although we do sometimes work with third parties as well. And in that, we're already announced that we're working with Novartis and Immune Design. So in our pipeline itself, the top and the middle lines deal with gene therapy products, the bottom with 5T4. And in ophthalmology, we have 2 products that are actually identical, although differently named. Retinostat is a product that produces both angiostatin and endostatin when injected subretinally. And we've been studying that in wet AMD for some time, and we have a presentation coming up at ARVO in early May, and that will also be one of the hot topics we were pleased to see. So we'll be releasing that phase 1 data then, and obviously working out what we're going to do next as a consequence of that. And then Oncostat is a different name. This is a topical version of the product, and we're using this because of its endostatin and angiostatin production to try and prevent the neovascular formation that is associated with corneal retinal rejection. And the more corneas you've had replaced, the more your risk of rejection goes up. And that's going to be ready for humans, we think, in the first quarter of next year. In the CNS, we've been working in Parkinson's disease as well, and I was interested in Steve Paul's comments at the first session in this room this afternoon. We've already published data last January on the 6-month and 1-year outcome of the 15 patients who received our product Prosavin. And in that, it's a lentiviral platform with 3 genes on it that convert tyrosine through L-DOPA to dopamine. And we'll be looking at the 3-year follow-up on all the patients, and 4, 5, and 6 years on the first patients treated. And there'll be some reference to that coming up at the AANS meeting at the beginning of May in Washington. You'll note from that publication, if you looked at it, that we saw some good improvements at 6 months and 1 year in the patients. We think there's some room for yet more potency in that construct. So our very smart preclinical scientists rearranged the genes so that we could increase the potency of dopamine production without increasing the volume of injection. And that product, OXB102, is going through its final testing now, and we're looking at getting that into the clinic next year. And one of our questions for ourselves is there room for both of these products, or do we have to pick one above the other? And then finally on this slide, we also have a 5T4 program. This is an oncofetal antigen that's found in almost all solid tumors. And we've done extensive work there, and we have 3 clinical studies going on at the moment. One in malignant pleural mesothelioma, one in colorectal cancer, and one in ovarian cancer. And some of those data will emerge during the course of this year, and one of the studies probably next year, maybe 2 of them next year, but 1 this year. And we're also working on a CARP5T4 project as well that's currently in the preclinical stage.
And maybe just flesh out that last one. What's unique about the CAR-T program?
I'm not able to tell you at the moment.
To be determined,
well,
to be disclosed.
To be disclosed, because obviously it's an extremely competitive field, and there's some IP challenges here that we want to make sure we capitalize on, and the opposite, make sure we don't screw up.
So you're not the only company working on lentivirus, but I thought it was very interesting that Novartis came to you for assistance with their lentivirus capabilities for their CAR-T program. Maybe talk a little bit about what makes Oxford Bio unique from that regard, and why Novartis did choose Ox Bio to work with.
Well, first of all, we're delighted that they did. You may accept that I never used to have any grey hair until we started negotiations with them, because it wasn't an easy one-step process. I'm sure Novartis were looking at several other alternative sources, and certainly they put us to test, and we met their criteria, and took time and worked well with them, and together, in fact, created the final agreements, and we're delivering on those now. We're very excited that they're in the CTL19 program, and it got off the ground just a few weeks ago, I think. So it's really a question for Novartis as well as us. We think we were the best able people to meet their specifications, and they had confidence both in the IP and the know-how that we've been developing by working on this ourselves. So when you're making your own product for yourself, you're extra careful. We've had 3 INDs open for gene therapy products in the US, and have established a good working relationship with the FDA in that regard, and I think that's helpful. We've been doing the same in Europe, because that's our home base as well. So the EMA and FDA relationships, I think, were also helpful in addition to the technical competence.
And with regard to the IP, presumably it gives you freedom to operate. Does it also potentially give you any economics on other lentiviral programs in development? How do you think about the applicability of your IP to other programs?
I'm not an IP expert, so I'm the wrong person to answer that question, but our feeling is we're confident in the IP we have, and obviously every company tries to strengthen its IP position with further filings, and we think we're doing the right things there. But always behind the IP as well, you have to have know-how, and there's nothing like putting the IP into practice and working through the issues, and sometimes you deliberately don't want to file IP there, because know-how doesn't have an expiry date.
Right. We had a couple of ophthalmology gene therapy companies using AAV present before you, and you're using lenti for the ophthalmology applications. What makes lenti a good vector for ophthalmology? What makes it unique? What can you do with lenti that maybe AAV wouldn't be able to do?
I think it's more than just an ophthalmology question. So I think in general, we believe that the lentis can, I don't think there's any doubt about this, can actually carry a bigger genetic platform. So depending on the size of the payload you want, you might favor lenti. There's nothing wrong with AAV if the gene fits in that regard. There is the ability for lenti to get into dividing cells, so you can more truly expect the one and done concept that I think someone mentioned earlier, which may not be as applicable to AAV, but I think there's space for both platforms. We just happen to have picked one and try to develop that as best we can.
And how do we think about other indications within ophthalmology? Are you looking to further diversify your portfolio beyond what you've partnered with Sanofi and what you've identified internally? And
yes,
what might
the next area, and I think we've announced it in the past, we've been working with the Mayo Clinic for some time on glaucoma, And obviously there are subsets of glaucoma. There's hypertensive, normotensive, and juvenile. And those are all areas that we're looking at, but I didn't mention that specifically on a slide today, just for the limit you gave us of 2, and wanting to follow a rule not to stick too much on a slide, so hopefully someone will remember something that's on there.
The Stat program I find very, very interesting in its novelty and approach. Maybe just kind of take us through that program, what you've shown in the preclinical setting, and what the unmet medical need is for a product like that.
I think the unmet medical need is, here you have people who are getting corneal transplants who, unlike the wet AMD population, tend to be older. Just about anyone of any age can need a corneal transplant because of some kind of injury or accident, and it often occurs in people in their working life. And whilst corneal replacement is a well-established procedure that goes very well for quite a large number of people, there is a significant proportion who get 1, 2, or even 3, or fearfully occasionally 4 quadrants of occlusion, usually due to new vessel formation. And if you've had one element of a quadrant growing new vessels, you're much more likely to have more over time, and so that will increase over the years. Even if it looks okay for the first couple of years, by about 5 years there's quite a significant proportion of patients who do get neovascularization and visual problems. And if you have a second corneal replacement, your chance of neovascularization goes up even more. So we think there's an opportunity there, because I think particularly having a gene product that produces endostatin, angiostatin, and I think we'll be showing some data of a very sustained effect in that regard when we get to ARVO. We've got a very interesting application for this product there.
The preclinical data seem rather striking.
Well, one of the attractions, and others have referred to this, of ophthalmology, you actually can see the target straight away, and obviously at the front of the eye you can absolutely see it in everybody. And we've done this work now in different species, and so it seems to be a consistent effect, and we're encouraged and excited. It is a project that we were able to persuade the British government to give us some money to fund as well, and Brits typically are tight, so it's not easy to get money out of the government.
This may or may not be accurate, but I feel like I've been hearing about Retinostat for a while. What's kind of been going on with the program? And I guess we're kind of now getting close to a new inflection point. So what have we seen that's encouraging that we should be excited about heading into the next data set?
If you followed the company, you may have heard that Retinostat was part of the package of 3 ocular products that we'd given to Sanofi, or sold to Sanofi some time ago. And last year they gave that one of the 3 back to us. But they did put a very nice statement. It wasn't to do with anything of the product features. It was due to their own internal conflicts, and I think that was perhaps in part related to the acquisition spree they'd been on, and found they had some internal competition. So the product came back to us. That may be why you've heard about it. And I think there's been some talk, well, if one partner's given it up, is it unpartnerable or not? We don't think so. But thankfully with the Novartis deal, our financial situation is quite different now, so our pressure to partner is somewhat less than it was. So the deal terms have to be different, and it may be something we choose to take forward ourselves. And given that it's a different approach, it's not a VEGF approach, there are multiple different things we can do. Having demonstrated what will be demonstrated at the ARVO presentation, I think we have a number of options of how to take it forward, and do we take one or more than one of those forward.
Maybe you could just elaborate on what those options are and how that should impact the way we look at and interpret the data.
Just conceptually, you could immediately think of going directly head-to-head against VEGF therapy. You could think of going with VEGF therapy to increase the proportion of people who get an excellent result from VEGF, because not everybody does. Some people get a partial improvement, but not as good as they would like, and some people fail to respond. Another approach could be reducing the frequency of VEGF injections, and that's not unrelated to the one I just mentioned. So both increasing the proportion and the quality of response, as well as reducing the frequency with which you need to give VEGF. And then the other options are looking at the role of angiostatin and endostatin in the conversion from dry AMD to wet. And so there's another area there, as well as I think it was the Avalanche speaker mentioned, central retinal vein edema, diabetic macular edema. Those are other possibilities that we could consider. But even with the Novartis deal, we can't do all of those, so we'll have to be selective.
We'll see if there are any questions in the audience. And if not, I think we get a little break and come back at 3:10 for a panel on adoptive T-cell therapies. So, Paul, thank you, and thank you for the other companies for joining and presenting.
Thanks, Josh.
Thank you.